neuroprotection in Parkinson's disease is really the holy grail. We really are facing a major issue. Several clinical trials have been performed targeting multiple mechanisms of degeneration in Parkinson's disease, but none of them were really successful. So what I will be doing is to try to do a post-mortem study of these clinical trials and try to explain why there are not successful. And I will cover 7 reasons. The first reason is that multiple etiology account for Parkinson's disease. For instance, there are some forms which are inherited forms, some are more environmental. And for instance, in my group, we have studied a very peculiar Parkinsonian syndrome in the French Caribbean, which is due to consumption of traditional medicine, including a toxic compound. And therefore, we suggest to perform clinical trial in more defined small population of patients with known etiology. The second reason is that the treatment is administered too late. There is a pre-symptomatic phase, and we suggest that clinical trials for neuroprotection should be started during this pre-symptomatic phase. And we have patients at risk which are carrier of mutations, but which do not express the clinical manifestation of the disease. And this might be an interesting population to test these clinical trials. The third reason is that the drugs are not delivered properly to the brain and to the very specific regions in which degeneration occur in the brain. So we propose to deliver the molecule in the right place. The fourth reason is that most of the clinical trial focus on the dopaminergic neurons, but we know that non-dopaminergic neurons degenerate also in Parkinson's disease. And my group has shown that a very small nucleus in the brainstem, it is called the pedunculopontine nucleus, is involved in gait and balance disorders in PD. So we should also take into account the non-dopaminergic lesion. The fourth reason is that the mechanism involved in cell death might vary during the development of the disease. So in other words, at the beginning and end stage of the disease, we might have different mechanisms. And for instance, at the end stage of the disease, we are facing a very strong neuroinflammatory process, which in fact might be involved in the self-perpetuation of the pathological process. And this is a very interesting target that should be analyzed. Finally, the different ways by which the neuron degenerate in Parkinson's disease might be additive, and therefore we might need to use cocktail of molecules. For instance, molecules targeting the altered protein processing and anti-inflammatory drugs. And brain environment is also changing in Parkinson's disease with changes in the blood-brain barrier, which might in fact also have to be taken into account. So we have multiple ways of improving the clinical trials, and we hope that this might lead to successful story for the benefit of the patients.